organism from many diseases. Immunity, we describe the immunity for the plant. Immunity in the plant can be classified into two types of immunity. The first is structural immunity, and the second is biochemical immunity. Also, we describe the immunity in the human, and we talked about the immune system. So we describe what is the immune system. Immune system is specific for the lymphatic system. The lymphatic system is a system which protects the living organism or the human especially from the infection by many diseases or the invasion of pathogens. What is the pathogens? Pathogens like microbes, spores, germs, which can penetrate the body. How the human body can protect himself from the invasion of these microbes according to the lymphatic system. Also, we describe some types like the lymphatic organs. What is the lymphatic organs? Like the spleen, tears, tonsils, thymus gland. Okay, all of these items we can talk about them in the last period. Today in this session we will complete the lymphatic system, and we start with the non-specific response. What is the non-specific response? What is this figure? This figure indicates the skin cut in the human body. When the skin cut, there is a wound cut in the skin. What happens if there is a wound in the human body? First, the tissue, like the epithelial tissue that is found in the skin epidermis, and you must to know that is the skin epidermis is a part of a squamous epithelial tissue. What happens for this tissue? If the cut happen, the white blood cell come out. This type of white blood cell called neutrophils, okay, and also another type known as monocytes and macrophage. There are three types of white blood cell: neutrophils, monocytes, and macrophage. These are come out from the human blood after infection or after cutting. What happens? These vessels or these monocytes, macrophage, and neutrophils engulf the bacteria and the damaged cells. What happens for the presence of, if the cut happen, the presence of bacteria, like the bacteria found everywhere, in air bacteria, in water, above our skin there is a bacteria. This bacteria causes the mast cells and the basophils to secrete histamine substance. What is the histamine? This is a substance found in the skin, allow us to compensate this bacteria. Okay, after the bacteria enter, like you can see in the figure, this is the bacteria. Okay, bacteria enter the skin. The mast cell engulf, okay, engulf means take it inside the cell, and the histamine granules secretes. This is the histamine granules. After engulf many of bacteria, what happens? Number 3, histamine granules increase the permeability, the local blood vessel, and the injured site stimulate the other immune cell, many cells to enter. After it enters the body, what happens? Number 4, the histamine granule causes dilation of blood or local blood, means the blood vessels become increased inside. So the flow of plasma to the site of injured increases and causing redness or swelling. Redness means the skin become red in color. This is indication for the histamine and the swelling and pain. Okay. After that, what happens? The white blood cell called the neutrophil and monocyte macrophage come to the site of infection, and the local blood vessel engulf the bacteria and damage it. So when the skin is cut or the bacteria penetrate the wound, what happens when the bacteria penetrate the wound or there is a cut in your hand? The bacteria found inside this. The white blood cell engulf this bacteria and take it inside the capsule and secrete histamine. This histamine granules make the skin become red in color and allow the another type of monocytes, neutrophils, and also macrophage to come out from the blood vessels. This is due to the dilation. Dilation means become wide. These cells come out from your body cells, and what happens? Engulf the bacteria. After engulfing the bacteria, all of these injured or all of these microbes can be killed without infection of the human. This is happen when the skin is cut, and this type of keeping the skin known by non-specific response. Okay, non-specific response means this bacteria, there is no certain type of infection or certain type of blood cells attack them. Okay, can be attacked by three types. Okay, macrophage, neutrophil, and monocytes. What is the acquired immunity? The acquired immunity is the immunity used by the body to resist the new pathogen in case of the failure of the natural immunity in getting rid of the pathogen, or resist the pathogens that have infected the body before. This means what? If the natural immunity inside our body fails, okay, to recome or remove this pathogen, what happens? We need another type of immunity known by acquired immunity. This acquired immunity may be from antibiotic, may be from vaccine or serum. This is another type of antibiotic. What is the immune response? Immune response, it is a series of specific defense mechanisms that done by lymphocytes to resist the pathogens. Means that there is another mechanism for defense. Defense means keeping the body away from pathogens. These lymphocytes resist the pathogen, means overcome or killing the pathogens to be suitable for the human. What is the humoral or antibody mediated immunity? Humoral or antibody mediated immunity means that the immune response through a certain type of white blood cell known as B lymphocyte. This B lymphocyte defend the body against aggressions of pathogens like the bacteria and viruses, and any toxin substance need to enter the body through the plasma or lymph and producing antibodies. This is when you infect, we will describe this slide, when you infect by a disease, what happens? This disease like bacteria, viruses, or any toxin substance, when enter the body, the immune response for the white blood cell, which known by B lymphocytes, defend the body against this microbe. If this cell fails to overcome the bacteria and viruses and toxin substance, what happens? The lymphocytes or the lymph producing another type of antibodies which make removal of this toxin substance. What is the mechanism of acquired immunity? The acquired immunity is done through two apparent separated mechanisms. First, or actually interconnected with each other. The first known by humoral or antibody mediated immunity, and the second known as cellular or cell mediated immunity. So there are two types of acquired immunity. The first type of acquired immunity known as humoral immunity, the second type known as cellular immunity, and these two types interconnect together or cooperate together to overcome the infection by many diseases. And you must know that the May or humoral immunity play an important role to overcome this, and by the action of the cellular help to removal the pathogen from the body. Okay, now we are going to see what happens by this. What is the steps of humoral body? What is the steps of humoral body immunity? Number one, attachment of the B lymphocyte with antigen. Antigen, this is coming from the bacteria or viruses. The first attachment means become adhere to it. Adhere means catch this antigen. When a pathogen enters the body carrying on its surface specific antigen, any pathogen invade the body, أي pathogen, viruses, bacteria, في زي like key. Okay, this key is suitable or known as antigen. This key needs a specific lymphocyte to recognize or to know what is this antigen, and attaches it by using immune receptors. These immune receptors found on the B lymphocyte. Okay, يبقى B lymphocyte contain immune response or immune receptors, while the pathogen contain antigen. When they attach to each other, what happens? The B lymphocyte can kill the antigen. Okay, the antigen bind with a protein in the B lymphocyte that is called major histocompatibility. Okay, or the complex known as MHC. MHC means major histocompatibility. Okay, this is the type of protein that when this complex resulting from binding between antigen and the protein transfer to the surface of B lymphocyte. Okay, you must know that the B lymphocyte recognize the antigen. After that, make a intermediate compound. This intermediate compound known as MHC, major histocompatibility compound protein. This protein susceptible for killing the virus or killing the bacteria by knowing the antigen and removal from your body. Okay. Also, this protein transfer to the surface of B lymphocyte. What is the role of macrophage? Macrophages at the same time engulf the antigen and digest it by their lysosomal enzyme into small fragments. Okay, you know that is inside the cell. There is each cell contain a type of lysosome. This lysosome in the structure of the cell, and it is found in large or major quantity in white blood cells. These lysosomes contain digestive enzymes which digest and engulf this microbe, and killing it means lysis. Lysis means kill the microbe, and after that it changes into small fragments and maybe go outside of the body with excretory substance or through the kidney, or store until it completely digested. Okay, now we are going to see what happens after that in these small fragments. These small fragments coming from the lysosome bind inside the macrophage. After it bind and formation the major histocompatibility protein, what happens to MHC? Then the complex resulting from this binding between antigen and MHC transfer to plasma membrane. Plasma membrane means the outside of the white blood cell of macrophage to be presented on the outer surface, to be presented on its outer surface. Okay? Now we are going to see what happens in this figure. The first, the role of macrophage in humoral immunity. This slide show what we said in the last slides. Number one, this is the macrophage. Okay, this is the lysosome. Okay, receptors. These receptors are found on outside of the macrophage. After that, what happened to the pathogen? This is the pathogen which carry antigen. This is the receptors of antigen. You can recognize it. After recognizing, take it inside the cell. And what happens after take the lysosomal or the lysosome combine with it to lysis. Lysis means killing it. Then it macrophage, after digest this antigen by lysosomal enzyme, this macrophage displays with MHC complex at the surface of the plasma membrane to go outside the body and get rid of this toxin or pathogen substance. Okay, what is the activation of the helper T cells? Helper T cells found also inside the blood. This is the TY cell. We recognize the antigen through the MHC protein that bind with it on the surface of macrophage. The helper T cells, TH means helper T cell. Okay, helper T cell bind to MHC, major histamine compatibility complex, through their receptors CD4. This is a type. This type of the receptor found on the outer membrane of the cell that present on the surface be converted into the activated helper T cells. Okay, so the T helper T cell or TH can be, after adding to MSH, form receptor CD4 to change into TY, TY named by activated helper cells. Okay, the activated helper T cells release protein. This protein known as interleukins that will activate the B cell, carrying on their surface the antigen linked with the MHC protein. After the activated protein become active, what happens? It produces a certain type of protein. This protein known as interleukins. The interleukins activate the B cell to carry this protein outside the plasma membrane. Okay, helper T cell, after recognized and after treated by the macrophage and presenting it on their plasma membrane, linked with the MHC protein. Okay, after linked, what happens? Production of antibody. This is the antibodies that against the microbe. These activated B cells start their work by dividing or multiplying to differentiate at the end of two types of cells. Okay, this antibody comes from the activated B cell after the activated B helper attack or combine with the protein. Plasma beta cell or B cell produce large amount of antibody which pass through the lymphatic vessel and the blood circulation to fight the infection. This is the pathway. What happens when infected by microbes? This the role of a lymphatic vessels to overcome this. Okay, what is the memory beta cell? Memory B cell, inside your body there are two types of cell. The first is a cell named by memory cell. What is the memory cell? Memory cell is a cell for identify the microbe when it enter again inside the body. When you infected by a viruses or bacteria or any type of pathogen, what happens inside your body? The lymphocytes and the beta cell or B cell become make some memory cell and put them inside the body or blood plasma. This memory cell, if you infected again, okay, يعني لو حصل infection in the next time, what happens? This memory cell produce the antibody which kills the microbe. Like what? Like the infection by measles. measles اللي هي الحصبة. لو حصل infection by measles, what happens? After the infection, the blood keep some of this memory cell inside the blood. What happens if you infected next time by the bacteria of measles? What happens? This memory cell come out and invasion this microbe or prevent this bacteria to enter the body again. Memory P cells remain in the blood for a long period of time, 20 to 30 years. This is for recognize the same antigen if it re-entered, okay, or removal the body again. Re-entered means become again, penetrate the human body. If there is invasion by this bacteria, the memory B cell can kill and prevent this infection to occur. Where they divide and differentiate into plasma cells that secrete antibodies specific for the same antigen, making a rapid response faster than the first response to the first infection. This is what happens inside this. The destruction of pathogens, microbes, means what is meant by destruction? Destruction means this pathogen can breaking down or there is no pathogen. How can invade this pathogen or destruction of the pathogen? The antibody that produced by the plasma cells will reach the blood circulation. Okay, after reach to the blood circulation, killing the microbes inside it. Through the lymph where they bind to the antigen that found on the surface of invading pathogen. This will activate the macrophage again to re-engulf this antigen, and this process continue for several days or weeks. Okay, this is if the infection occur for the next time. This is what happens. Okay, you can see this is the bacteria. Okay, bacteria, antigen fragment. Okay, macrophage present or presenting the cells of antigen on its surface. After that, what happened? Okay, clone or a large number of memory B cells, and this is clone of plasma which secreted antibody molecules to invade or overcome the infection by this disease. Cellular or cell-mediated immunity. Cellular immunity, it is the immune response that done by T lymphocytes through the receptors which found on their membranes and give them a specific response to the antigens. Okay, this means that there is another type of immunity without humoral immunity. There is a cellular immunity. Cellular immunity, it is the immune response, and this is for the T lymphocyte by which or through which the receptors found on their membranes and give them a specific response. Okay, what is the specific response to antigen? Each T cells produces during its maturation a specific type of receptor which is specialized to its membrane. Therefore, each type of these receptors can bind to a single type of antigen. Single type of antigen. Dear students, I hope you enjoy our time today. We finish the immunity, humoral immunity, and we will continue the T cellular immunity in the next session. Thank you for your watching, and goodbye. See you again.